Okay, so 11 o'clock, let's have a class. We will move on to the exam 2. Exam 2, we will talk about the pathogen caused the disease. We'll focus on the bacteria. So exam 1, when we finish, is those basic knowledge. Then exam 2, we'll talk about is bacteria, pathogen, human pathogen, cause the disease. Here we also will talk about the antibiotics, and at the end we'll talk about the control of microorganisms. Here is something we want to let you know. When we talk about bacterial pathogen caused disease, we'll talk about 3 to 4 lectures. We'll introduce you about like 12 to 15 pathogens. And these pathogens is, we're going to talk all these characteristics related to disease, all kinds of things. It's relatively very comprehensive. So you're going to take the notes, and also I will suggest you make a flashcard for all these pathogens, the characteristics. And also we will be practice lots of the case study. Because in the exam 2, we're going to put lots of the case studies there to you to figure out which case is caused by which pathogen. So give you some of the examples. On the eCampus, I have the slides, but also very important, I have all these after-class reading material. This is I've been teaching for 11 years, and from different textbook has been concluded with these notes. So you should read after the class and over the weekend to be prepared, especially these case studies. We're going to mention some of them in the classroom when we get a chance. But you have to read about these first. This is basically is the outline and the material what we're going to talk. And it's a very good conclusion material. We have after class reading 1, 2, and 3. You definitely should do. And give you another example. So, for example, we're going to talk about the corner bacteria today. Then, in this segment, sorry, I didn't move that. In this segment, we have one paragraph talk about corner bacteria. That you should read. And we have a case study related to that. That is possibly will be in the exam, or similar thing will be in the exam. And then you should read. And next class we talk about Klebsiella, and you should read. So Haemophilus influenza we're going to talk about today. Here are the conclusion and the case study. So that is every pathogen with a story behind it, and then we go these one by one. So that's how we're going to learn. Then when we finish, we're going to talk about antibiotics. So we talk about the control of the microorganism. Okay, so that's how it goes. So let's talk about these first. The first section we will talk about is upper respiratory tract disease. So before that, we go a little bit easy. We talk a little bit about the respiratory tract, the anatomy. So you can see the intersection between upper and lower respiratory tract is epiglottis, is right here. That is a soft bone. And you know that, even you were very young, your parents will tell you, when you're eating, please don't talk, please don't laugh. Otherwise the food will go to where? Not go to the stomach, will go to the glottis, or go to your lung area. Because the epiglottis, it's a soft bone. Ideally, it should be closed when you're eating. However, if you're laughing or talking, it will be opened, or sneezing a little bit out, and then the food could be go to the lung. You could be choked. So that's important places, and it also the intersection between upper respiratory tract and the lower respiratory tract. Then you see the esophagus basically is connected to the bronchi and go to the lungs area. And you're also going to see a very important place here is called the tonsil. And some of you, the tonsil has been taken out. Because tonsil is a good reservoir for the bacteria to stay there. And then they can translocate it when the weather or condition changes and cause all types of the problems. And also the nasopharyngeal tract. You know. And we're going to have some test which is using nasopharyngeal swab to swab the sample from there and to see what type of pathogen it is, and it usually has to determine on the transport media go to the reference lab. So this gives you a very brief overview of the respiratory tract of what it looks like. And this slide basically is the outline what we're going to talk. Some of them we are going to talk relatively easy, some of them we'll go much more details. So let's say the table, you have the colon and also the row. The colon is the disease. And on the right side of the colon, you see the microorganism corresponding to each disease. And the difficult thing is, sometimes the same disease will caused by multiple different pathogens. So for example, among pneumonia, you can see they listed like 6 or 7 or even more different pathogens will cause pneumonia. So this is something you have to learn. And also meningitis. We have 3 pathogens caused by meningitis. So keep in mind, it is not one by one or one corresponding to one. Multiple pathogens could cause the same symptoms or the same disease. So we will talk real quick. Corynebacterial diphtheria cause diphtheria. Bacterial pertussis cause whooping cough. Pretty much will be next week. Then pneumonia, you have Streptococcus pneumonia, Staphylococcus aureus, Legionella, Mycoplasma, Klebsiella, and pneumocytosis. Then streptococcal infections, Streptococcus pyogenes. This is we already mentioned a couple of times. And today we're going to talk about the meningitis. We have a Haemophilus influenza, Neisseria meningitis, and Streptococcus pneumonia. And the last one we already mentioned, tuberculosis, Mycobacterial tuberculosis, started by Bob Koch using acid-fast staining to do the pre-clinical determination, because Mycobacterial tuberculosis, the doubling time is very long. It's about 12 to 24 hours. So we're going to pick some of them. We're going to talk today, followed we'll talk next week, Tuesday and Thursday. Something very briefly mentioned here. Pneumonia is a big topic. Some of them will go very much detail, but there's something I will be briefly mentioned here. Mycoplasma pneumonia and pneumocytosis, these two we call it a walking pneumonia. What this means, walking pneumonia? This means that symptoms is relatively weak or mild symptoms. You have like a little bit sneezing, slightly coughing, maybe 37.5 degree Fahrenheit, those lower temperature, fever, and it will be disappear by itself without any medicine after you rest for a couple of days, 5 to 7 days, or sometimes 3 to 5 days. That's called walking pneumonia. The one what we hear, I want to talk a little bit more, is a Legionella pneumonia. So what is a Legionella means? This happens in 1970s in the Army Convention in Philadelphia. So after the Army had a convention, have a parade, they go to take a shower. And the second day, 80% of the people have pneumonia. Because the Legionella is easily to be transferred in the aquatic system, especially like a shower. Like even the heated air conditioner, all those things, related to the water. So But Legionella ammoniae is not going to cause death, or the rate to cause death is very low, so it's not that being a high risk. But in our municipal drinking water, we do require the Legionella has to be tested. And our standard is zero tolerance for Legionella species found in our municipal water, the drinking water. Okay, so this is, I'll be very briefly talking. So let's go detail. The first one, corner bacteria diphtheria. Corner bacterial diphtheria, I want to make sure the spelling is correct. Genus name, species name should be italic. So where this comes from? C-O-R-Y-N-E is corner. This is from Greek word, means club. So corner bacteria diphtheria. Remember, we do gram stamp. We had a question. What are the three major items we have to remember and record it? Is gram reaction, morphology, and arrangement. And don't forget about that. That is the three basic information when we go over every pathogen. So Corynebacterial diphtheria, it is gram positive, large shape, club arrangement. So what means the club arrangement looks like? Could be this, could be like that, could be like this. Sometimes they say this is like a Chinese characteristic too. That means the club. Looks like people are stacking together. And lots of the time, you can see, if you look more carefully, inside there is a dots. And what is that? This is called inclusion. The inclusion, usually, it is a storage of some nutrients, for example, a phosphate. It's very tiny. Look at this one. The slides tells you gram-positive, lots, and the club shape. And we also call club shape is pali-size. When you look at this, did you feel about when you guys do the lab, in the labs 2 and 3 do the simple stain. I said this is E. coli. It's labeled as E. coli. Then we use microscope to do the observation. Did you feel about it looks like that? Some of my slides looks like that. So I said that by the time, it's possible it's a corynebacteria which mixed with E. coli in your lab as a sample. Maybe not a diphtheria, because there's a different corynebacteria species, but looks very similar. And this is underneath phase contrast microscope. So it's more clear how they looks like. Purple, it's gram-positive because crystal violet color. Okay, that's corynebacteria. We want to talk about thing directly goes here. So these are the characteristics. Corynebacterial diphtheria. The high-risk group is immigrant kids. I've been teaching this for 12 years. These days I shouldn't talk these things because it's really sensitive, but from political standpoints, but the CDC data has been showing in a long term, the immigrant kids is a high-risk group. Why? The reason is in the United States, we have a very effective vaccine for that. And that vaccine, everybody get a shot. I just got a shot like a month ago. That's called DTaP. You got that, called TAP or DAP, whatever they call it. That's a vaccine you get when you were very young, like 2 months. Then have to do the immunization like 6 months, 8 months, 12 months, 15 months, then stop. Then after like 5 years old, and then when you become adults, they're going to have 10 years, have to do a shot. What this means? D. Bacteria, diphtheria. T. Clostridium tetanus. We'll talk next week. A. Cellular. P. Bordetella pertussis. Cause whooping cough. So 1 shot, 3 jobs. But why we have to do a booster? Which means you have to do multiple shots during the lifetime. The reason is this DTAP, it's a toxoid, which means it's a toxin-like chemical. It only activates T cell line and not very much for the B cell line. So when the T cell line comes out, they have a universal protection system. B cell usually have antibody, but this line they didn't go. So in order to have an active immune in your body, we need to give the booster. So that's the reason have an immune scheduling for this one. So in the United States, we have this vaccine. So we don't have a problem. Now, I never heard about, or very few heard about the people that are born here. You have a problem because you go to daycare, you go to elementary school, you go any public and private education institution, you're required to have that. So like me, when I came here to study 20 years ago, we have required to have the immune due before we can register classes. One of that is DTAP, another one is MMR. We're going to mention sometimes later. So just want to let you know, in the United States, there's no problem. But for the immigrant kids, people from South America, let's say Central America, let's say from El Salvador, Mexico, Honduras, those places, if they didn't get an immune for the vaccine, very easy to have diphtheria. What's the diphtheria symptoms will be looks like? If you have a human being like that, a very sick ball neck. Look at the picture, that's what it is. Why the ball neck will happen is swelling, severe swelling of lymph nodes. Certainly it means the immune system is highly activated. The second thing, the nurse usually will say, can you please open your mouth? Then of course the kid just wow, 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 wow like that. Then when you open your mouth, you say, what happened? In this tonsil area, remember we said that this is a reservoir for bacteria. It will have a pseudo membrane. A fake membrane. Looks like there's a membrane, but you use cotton swab to touch. It's very soft. It's easy to broken and it starts bleeding. That's what the major symptoms looks like. And this pathogen usually is for kids, 2 to 5 years old, and older people more than 40 years old. So that's what the symptoms looks like. Now the question will be, what is this bacteria cause? Why it will have this problem? This is a diphtheria toxin. Okay, this has a diphtheria toxin. This is a toxin. In other words, we have a genome name, we have a species name, and the species name is coming from a toxin. And where this toxin comes from? This toxin coming from a phage called a beta phage. So what happened is, let's say I have a bacterial cell. Okay, I have something is coming. The phage looks like this. It's a bacteria. They have a capsid, which is a protein at a coat. And the inside has DNA or RNA. Now usually here is a toxin. And this is also we call it an exotoxin. So what means exotoxin? Exotoxin is heat resistant, slightly heat resistant, compared to endotoxin. Remember we talked about LPS. Lipopolysaccharides for E. coli, Salmonella will have endotoxin. And endotoxin, you heat it, it will be killed because it's called heat liable. Exotoxin, it is heat resistant. I will just say slightly. Of course, you cooking it, it will be died too. So the exotoxin, then the exotoxin find a bacteria. One day, they attacking it, or we say invade it. They stay there as like a parasite to the host. And this corynebacteria has a bacterial genome, which is a sequencing. And then what happened is this toxin integrated into the bacterial genome. And then many times of the replication and the reproduction, and we call it microbiology, evolution, many years, it stays there. And then become an exotoxin. That's why it will be there. It's very similar like endocytosis for the human being. So remember, we have a lot of cell machinery in our body. Why we have? Because a long time ago, they think this coming from a bacteria. A bacteria is engaged with a human being like a host to the parasite, and they stay there. This is the same thing. And we have even a terminology for this phenomenon we call lysergeny conversion. So that's why it will cause a problem. Now this toxin, why they do, they will to prevent the DNA replication to RNA transcription. And basically they are attacking elongation factor 2. We'll talk about that. And by the way, when they go to the cell, they usually will have an endosome comes out. So that's originally people doing some of the research. Now later on people find this toxin have other things. It is a typical AB toxin. It has 2 subunits. So we're talking about the toxin. It looks like a protein. It has 2 subunits. It's an AB toxin. A is, we call it a toxin domain. The toxin domain basically causes inflammation. So what is the inflammation means? You have a heat, you have a red, you have a swelling, and you have a clotting there. That's called the inflammation. Now what the V domain, that is transmembrane domain. Transmembrane domain will has a problem. Since it's transcell membrane, what it cause? It will cause bleeding. Because of that, you have the pseudomembrane, which is soft and cause bleeding. So that's what is that toxin comes. Now the toxin is very interesting. This bacteria is localized. It stays on the tonsil area. So this pathogen itself is localized. It's not penetrating. So when we talk about streptococcus pneumonia, all those, it's penetrating. It goes everywhere. It's from the upper respiratory tract to go to lower respiratory tract. It goes any places. This is localized. But the toxin goes everywhere. So that's caused a lot of the problem. Now we look at these kids, the picture, you see the cedal membrane system, you see the ball neck. This is some early images, which is early signals. Later on, it will happen neurological symptoms. What are the neurological symptoms? Basically, we say it is double vision, difficult speak, and difficult swallow. And it all comes from, which is a toxin. It's an exotoxin, comes from beta-phage. So we can go back to see here. This is the diphtheria toxin we talk about. We had these two symptoms. And I said in the United States it's not a problem because we have this table. Look at the top. Remember the first shot of DTAP at 15, 18 months. And after that, 4 to 6 years you have to do. And when you become adults, every 10 years. And these have the immunization for others. We're going to mention this later when we have. This is basically, we talk about the mode of the diphtheria toxin. The picture showing here is the AB domain. The toxin domain and transmembrane domain, when they go to the human body, it will have an indosome. The indosome going to generate. It usually is a slightly low pH, like pH 5.0. Then the A and B domains, they will be separate. And the A domain, we call it an inflammation domain. They will prevent the elongation factor 2, which is prevent the DNA transcription or translation. A little bit about replication, they're all connected. And the B domain is a transmembrane domain. It's caused toxin. It causes bleeding. And then the B domain, when they come out, finish the job, they come out, and they're going to connect it with a new A domain, and it comes back. So that is what it happens, the mode of the diphtheria toxin. Now we want to talk about how we do it. How we do the isolation from the bacterial standpoint. Once you go to the hospital, you have all these symptoms. Now right now, of course they have the rapid testing for the exotoxin. So usually they use a cotton swab and go to the testing kit. They put it in. After 1 minute, they tell you it's positive or negative. Very similar to your COVID-19 test kit. But that is only tells you have the toxin. It's not for isolation. But how are we going to do that isolation? The cotton swab when you usually have to streak plating onto a transfer medium. Bacterial medium. And before that, we need to do a presumptive testing. So we're using a special selective and differentiated media. We call it called cysteine telluride blood agar. It is an enrichment media. It is also a differentiated media. On cysteine telluride blood agar, you will see those black colony. Now, of course, the black colony will be faded because of reducing of the salt. And later on, when they're exposed to the oxygen, it will be re-oxidized. So once you have a black colony, these black colony, you're going to pick it. And then transfer bacterial media here. Leaflose transport medium. And then this media, you basically go reference lab. I don't know how about here. I used to work at Kentucky. In Kentucky is in Lexington, close to the University of Kentucky Medical Center. They have the reference lab. The reference lab will do a confirmation. And if it is determined 100% it is Corynebacterial diphtheria, this need to report to CDC in their database. And they also further will do a molecular classification to see what is the genotype or serotype or these things. So that's what the story for Corynebacterial diphtheria. Started from the characteristics of this pathogen. The risk group is immigrant kids because they don't have a vaccine. The typical symptoms is ball neck, swelling of the lymph nodes, with membrane at the tonsil area. It all caused because of the toxin, the exotoxin. And the exotoxin originated from beta-phage. We have a terminology, we call it a lysogenic conversion. And very important, this is the AB toxin. So it says toxin domain and inflammation domain and also the transmembrane domain. We will mention several pathogen very similar to Corynebacteria. It is also the AB toxin. Now what we should study is all this information obviously is in the notes what I give to you the after class reading that you should highlight it. And for yourself, you also have a flashcard which is talk about. So the back of the flashcard, you should talk about the characteristics of the pathogen. At the front of the flashcard, you should give one of the case study. The case study I'll give to you later on when we do the review. You should stick there, and then you can very easy in the morning before the exam. You go ahead to take a look. It's very good. Membrane or networking system in your mind about all these pathogens. Last thing I haven't mentioned, what we should do if people have Corynebacterial diphtheria. Obviously, we cannot do the vaccine again because it's for prevention, not for treatment. If anything happens, remember it's a toxin, so you have to give antitoxin first to neutralize the toxin. But it's a bacteria. Of course, you need to get antibiotics. So this has to be double treatments with antitoxin and antibiotics. Okay? So this is for diphtheria. Next one is a big topic, is for meningitis. This pathogen causes meningitis. As a lot. So we're going to mention several. Start from Haemophilus influenza, go Neisseria, meningitis. Some of them, we talk details next week. So let's talk about the meningitis. We first of all will think about what's the symptoms for meningitis. Let's go from here. What's the symptoms for meningitis? Imagine if people have meningitis. What will be the symptoms? Of course, you have a headache. You pretty much, some people have vomiting. Some people have photophobia. Scared to light. But there is one thing you should always remember, and you should always do not dismiss it. Tough neck. Lots of people think it's a muscle problem. If you have a tough neck, has not been recovered for a couple of days, that could be the meningitis. And this is typically in undergraduates, which is young people, less than 25 years old. So these are the symptoms, are very typical symptoms for the meningitis. Now what is meningitis? It is inflammation between brain membrane system and cerebral spinal cord. Now obviously they're going to generate the cerebral spinal fluid, so-called. That's the inflammation will happen. Because of the inflammation, you have these symptoms. That's the first thing you need to know. Second, we just said lots of the pathogen will cause meningitis. And often the time, it is age dependent. Which means different age group have a more related risk pathogen. If you are 0 to 2 months, this usually is a newborn baby. The meningitis is caused by Streptococcus agalactiae. This is called Group B Streptococcus, GBS. We'll talk next week. What is GBS? If you're 2 months to 5 years old, the pathogen is HIV, type B Haemophilus influenza. We'll talk this real quick. If you are 5 years to 40 years old, that will cause Neisseria meningitis. And this typical symptom is a tough neck. If you're more than 40 years old, you're older people, surprising, Streptococcus pneumonia will cause meningitis. Although it says the species name is pneumonia. This is age dependent. So, we're going to talk about this guy immediately. Type B Haemophilus influenza. First of all, this guy, the genus name is confusing. It says influenza. So looks like it will cause flu. No. It will cause mild symptoms of flu-like when they first did the research. However, it caused a big problem, much worse than flu. Or much worse than walking pneumonia. It is caused meningitis. This pathogen is the #1 childhood disease in in the United States, before the vaccine comes. Now, obviously, we just showed you, right now, it should not be a problem. We had Haemophilus influenza type B, very similar to diphtheria, very similar to DTaP. 2 months, 4 months, 6 months, 12 months, you have to do a multiple shots, which means a boosters. But before that, it's #1 childhood disease in the United States, cause meningitis. What's the characteristics of this bacteria? We're going to talk more deep for this one. This is gram-negative, tiny Coccobacillus. Like that. Looks like a cocktail, looks like a bacillus, looks like a lot, we call it a Coccobacillus. This bacteria is very interesting. Remember, we talk about the bacterial media. And I said it is important, you should remember. Because we're going to talk about these today and next week. In the clinical area, there are 3 other plates is important to do the predetermination, which is we did in the lab, McConkey agar, followed by blood agar, last chocolate agar. Haemophilus influenza does not grow on McConkey agar, although it's gram-negative. Does not grow on blood agar, although blood agar is an enriched media. Very special. It only grows on chocolate agar. What this means? It is a fastidious bacteria. Very picky. So that's ironic. It's difficult to grow in the lab, difficult to determination, but it's a pathogen. It's caused severe problem. And more interestingly, when people do the research, what they find somehow really interesting is, if I had a neutral agar or a TSA agar, I put some factor there. I put X factor, I put an X V factor, we put a X and a V. So what is X means? That's hemi. What is V is NAD, nicotinic adenine dinucleotides. That's an electron transfer. We will talk about in exam 3. What they find, they did not grow here, they did not grow here. They grow only once they are coexisting X and V factor. So which means when they grow, they do need it, heming and NAD. Now why they do that? Because there's a lot of Haemophilus. They find that there is another pathogen called the Haemophilus parainfluenza. This parainfluenza can grow only with X factor. And then they dig a little bit more, they find the parainfluenza will cause endocarditis. That is inflammation of the heart valve. But very special, this Haemophilus influenza must need X and V factors. And it's also very interesting because lots of the patients, they're not going to only have one pathogen isolated. They have a lot of others. Then one day what they find over blood agar, over red blood agar, if you grow in Staphylococcus aureus, and they find Haemophilus influenza can grow, but very tiny. This is called satellite phenomenon. So these are the 2 ways, are very easy to determine Haemophilus influenza in the clinical area. And makes you get a final, like a final confirms, this will have the meningitis. So that's a very interesting pathogen we talk about. And this is the first time we talk about the Coccobacillus. But very interesting, lots of the human pathogen, when we go more and more, 90% of those are Coccobacillus. They are tiny, they are also hard to differentiate sometimes. That's why we need those molecular methods, biochemistry methods, and also always remember those 3 agar are important in the clinical area to do the first preliminary determination. Okay, so that's Haemophilus influenza. We want to talk about something more is Neisseria meningitis, because it's related to the undergraduates, so we want to talk about that. Neisseria meningitis. This is another pathogen, which is coming from Neisseria. Neisseria is very special. You look at the picture, you get the sample, you do the gram stain, it's gram negative, it's Coccobacillus, it looks like a lodge. The morphology is okay, but the arrangement is very special. What it looks like a coffee bean. Did you ever make a coffee using the coffee maker originally? You buy a bag of coffee beans and you smash it. That looks like a coffee bean. So clinically what they say, this is a gram negative Coccobacillus and also diprococcal. Coffee bean shape. And surrounded by white blood cell. That's a Neisseria. Neisseria has one species is meningitis. Neisseria has another species is also famously for risk. Is Neisseria gonorrhea. It's a sexual transmitted disease. Number 2, sexual transmitted disease pathogen. Number 1, obviously, is syphilis. Number 2 is Neisseria gonorrhea. We'll mention later on. Neisseria meningitis have a different serotype. A, B, C, W, 135, and we also had a Y. In the United States, type B responsible for 60%. So this gives you some of the issue at the early time. Before 2010, in Connecticut and California, the university is required undergraduates to have the vaccine before they can register for the class. The vaccine is called MPSV4, MCV4. It's called Menumine. There is a commercial name called that, called Menumine. Menumine, or the problem is, in the United States, 60% of the chance it's a type B. The old Menumine vaccine can only cover ACW135 and Y. We're going to be flee away for the type B. So there's a big debate by the time whether we should require undergraduate students to have this vaccine. Because it's only have less than 40% of the protection rate. It sounds ridiculous because you get or you not get, that's natural chance is 50%, and you have the vaccine, you only have a 40%. So clinical areas, they have a big debate about that. Until 2012, they get 600 patients nationwide, isolate their type B, got the polysaccharides, and got the capsule. You can look at the capsule, how heavy it is. You can see the yellow one, that's a capsule. It's like a shiny, they get the capsule. Then they purify the isolate. They get a newly developed vaccine. 600 patients, they only get an effectively one, successfully isolated is only 25. Very few, it's less than 30%, less than 40%, they get it. And then they purify, they get the newly developed vaccine, start from 2015, it could be covered with all these. So recently, in the American history, they do have a big development for the Neisseria meningitis. It happens for a person's name of Ryan. I forgot the first name. He is undergraduate at Indiana University, not far away from here. So he's an undergraduate, biology major. One day he had a tough neck, so he had to take a break. He take a break and sleep, very, very drowsy and very, because of lethargy or something. The people, his roommate find that it's not normal. So, but you can take a break another day. Okay, he take a break another day. Then after 48 hours, it still looks like that. Then one of the roommates said that you need to go to the hospital. Then they go to the hospital. And the doctor, from this very limited amount of the cerebrospinal fluid, they get this Neisseria meningitis. And the doctor says, if you're coming another hour later, you could be dead. And later on, Ryan has been worked really hard after recovery because himself is a biology student. So later on, he graduated, worked at the Indiana CDC to develop these vaccines. And one of his work contribute is to isolate those type B pathogen and get those newly developed vaccine. Obviously, it's collaborated with other institutes. So just want to let you know, long time ago, this is a big issue. For the undergraduates in the crowded area, in the bar, in the library, or in those like a party area, sometimes people go there, then go back, have a tough neck, and then they just don't care. They think it's only the muscle hurt or just take a break. But if you delayed to see the doctor, it could be a problem, because obviously that might be a meningitis. It is more severe symptoms. Okay, so some story behind that. So meningitis here, if we do some of the conclusion, we'll talk about Haemophilus influenza. We also talk about Neisseria meningitis. But where we get the sample? You're not going to dig a hole in your brain to get the sample. Obviously not. So meningitis, we basically get cerebrospinal cord. We get cerebrospinal fluid. We get that sample. Then we go ahead to do the isolation. Often the time we have a rapid testing kit. We can know the multiple bacterial strains, whether it's positive or negative. If we are presumptive positive, we will do a further determination. Then use some traditional methods, and sometimes with the PCR methods to do the further identification. Again, this has to be reported to CDC, then the database will be record that to see what's exactly the species it will cause. Sometimes could be detailed to the genotype and also to the serotype, especially these days. You could get a whole genome sequencing. So what happened is, if you have a colony here, that's why the isolation of the pure culture is important. This colony tells you lots of information because it's a pure culture. You could go ahead to do the genome sequencing, which means you know the ATGC, all those sequencing. Then you go to the database, you will know exactly what it is. There has been developed like that. In the older time, people cannot do that. They can only do shotgun sequencing. All they can only do is PFGE, which means running the gel electrophoresis. Then compared to the database, that gives you like a 90% matching. They have a lot of rules. They have like 3 bands rules. Or your database have so many bands, your sample have so many bands. If you mispair only 3 bands, it will say 95% match. Those are still like a presumptive determination, but right now you could run the whole genome sequencing. That's called the next generation sequencing. You know all the gene base very well, so you know what it is. It's really fantastic for these type of the biotechnology work. Okay, pertussis. When we finish this one, we're going to end of the class. Okay, pertussis. This is one usually for child. Another one for child. That's caused by Bacterial Pathogen-1. Okay, this usually happens is people, newborn kids, 100 days. So in Asian countries, in China, we call this a 100-day cough. We don't call it here. Which means roughly 3 months is easily to get if you don't have a vaccine. Because, I already removed because we just said this is Bacterial Pathogen. That is a P. So this vaccine can also immune the Bacterial Pathogen. If you don't have the vaccine, a 100-day cough is easily to happen for the newborn. What is a special for the COVID? This is called a whooping cough. What is cough? Looks like that is you cough, 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 then whirl, then cough, 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 then whirl. That's called a whooping cough. Which means you had a productive cough with respiratory tract gasp. That's what a whooping cough looks like. It's a very typical symptoms for that. Now, obviously, if you do not give the treatments, it will be further developed, could become a neurological symptom too, because Bacterial Pathogen, it is also an AB toxin. Now, why they will have a whooping cough? This is kind of related to the respiratory tract, the cilia related to that. Said very simple, the mechanism behind that. You should have ATP generate. Instead of you have ATP, you have CAMP, cyclic adenine monophosphorus. That is not good. Once you have this comes out, it looks like the energy money is blocked. We'll have the respiratory tract gasp. We'll draw the structure later on in the exam 3, then you will know very well. So that's what happened. Now, how are we going to do the determination of this one? The determination is very special. It is interesting is let's say we have a kid who is get a whooping cough. We know to get a sample from the spit. How we do? We're using a very special swab. We call it a dacron swab. Usually the swab is cotton swab. Here, very special material called dacron swab. Because they do a screening test, they find that the general cotton swab will not have a good recovery, followed by PCR methods. So they're using a special one, is a dacron swab. Now how they do it? They first are going to measure the distance between the earlobe and the nose. So they're going to measure it. And then the dacron swab is going to go inside. Now how deep it is, half distance between earlobe to nose. That's how depth it should go. And then they took out the dacron swab. This also need to go to the reference lab. And then they run the PCR, the polymerase chain reaction. They can do a very rapid determination, is that Bacterial Pathogen. Now, obviously, basically, the family doctor will look at your immune system, immune recording first. If you do not have this guy, then very possible. That's why we said it's always happened for the immigrant kids that came to the United States with the parents, and they did not do the immunization process. So of course it will be easily to be contaminated. So here you can also see it has developed steps, 4 to 14 days incubation, and followed by those cold-like symptoms, and finally will be neurological symptoms. Now, obviously, this is a pathogen, bacterial pathogen. If you really have it, you can only do is do the antibiotics immediately. The challenge is this is also gram-negative bacteria. So gram-negative bacteria, I already said that using antibiotics sometimes is not very effective because they're very thin layer of the peptidoglycan. If it's really effective, then E. coli 0157H7, it will not be a problem. You can eat those undercooked beef. After that, you have a vomiting, just give you a shot to solve the problem, but not really. So keep in mind about that, the immunization, the vaccination, it is important to prevent these disease at the early stage. Now, obviously, with recent news, you can see everything comes out. So we have been saying about the vaccine, the coronavirus vaccine have some technical issue behind it. Those are some of emergent pathogens we don't know, so we have less research. And also we didn't go through a very detailed verification process, validation process. So those things happen. But these well-developed vaccine should be all very good to prevent these infectious disease from the history standpoint. I can only say from the CDC history recording standpoint, it is really helpful to prevent these human pathogens. Before end of the class, you see this before. That's why we have here again. This tells you the blood agar is important because it's an enrichment media and also the differentiated media. Remember alpha, beta, gamma hemolytic, and we will talk about this next week. And we will focus on the blood agar, talk about streptococcus and staphylococcus, especially streptococcus. There are lots of the topics we can cover. Okay? That's good.